Welcome to Module 2 of Immunology, Antigens and Antibodies. Today we're going to define the terms antigen and antibody and how they work together. We're going to look at the characteristics of each of the 5 immunoglobulin classes involved with antibodies. We're going to be able to draw and describe a typical immunoglobulin and molecular structure, which you are going to be doing in Lab Week 2 as well. We're going to name the 4 phases of an antibody response, describe a primary and secondary or amnestic response, and also describe the method of production of a monoclonal antibody. First, we're going to start with antigens. I'm going to give you an example just to get us started here. A lot of you guys have had to have vaccines before you could start the MLT program, or you've had to have them as a child. And how this pretty much works is if you are wanting to have an immunity to a certain disease, we actually have to take and lightly infect you with the antigen from that disease. So it may be an actual attenuated virus, kind of like the flu vaccine, or it may be some type of a toxin that a bacteria or virus could produce, kind of like the tetanus vaccine. Either way, we're providing the body with these antigens for which to make an immune response. So an antigen in general is a substance that stimulates the antibody production and has the ability to bind to an antibody. Once you've been exposed to those antigens from that vaccine, your body's going to create all those antibodies to help fight those off in the future, which is why we would get a vaccine to begin with. The antigenic or immunogenic refers to the capability of provoking that immune response. Foreign substances have to contain those antigenic structures. We call these antigenic determinants or epitopes. So that is the part of the antigen that the antibody would bind to. We do have some antigens that are really important in the body as well. Obviously, there's foreign antigens like I just talked about, like the tetanus and the flu and things like that. But we also have antigens that make our cells ourself. Histocompatibility antigens are an example of that. Nucleated cells such as leukocytes have cell surface protein antigens that provoke that immune response when put into someone else, which is known as an allogenic individual. But having part of this major histocompatibility complex is what tells our body it belongs to us. Okay? So my body knows that my kidney is mine because of the major histocompatibility complex and those antigens. It will not bother those. But if you were to take Sally Sue's kidney and put it in my body, it would have issues with that because we don't have the same major histocompatibility complex. So the body's going to start making antibodies against those antigens, and we're going to have rejection happen. We're going to have a lot of discussion on that towards the end of immunology. We also have autoantigens. These are kind of an undesirable consequence of the immune system. Your body can sometimes get a mind of its own and make antibodies against things it probably shouldn't, such as antibodies against organs or hormones. An example is thyroglobulin antibodies. A lot of people who have thyroids that are low functioning, it's usually because the body decided to make antibodies against the thyroid and attack it. This starts to destroy the thyroid, and therefore we have low thyroid function. So that's a very common example, but there are other ones as well that we'll discover as we go through. Here's some other antigens of importance that you've probably heard of. If you've ever donated blood or known anybody who's received blood, this blood group antigen picture you see here is of utmost importance. So we can see that if you have type A blood, there's a lot of type A people out there, you have these little pink, and they're not really pink, A antigens on the outside of your cells. If you're type B, you've got B antigens. If you're AB, you've got both, and if you're O, you don't have any, okay? So what happens is your body, if you're group A, makes anti-B antibodies. So what would happen if you gave a group A person group B blood? The anti-B antibodies would attack those antigens and destroy those red cells. This can be detrimental and life-threatening, if not cost someone their life, because bursting all of those red blood cells causes a high leach of potassium and cause heart failure. So it's a big deal, okay? Same thing if you were to give an A person, or sorry, a B person, A blood, because you would have anti-B antibodies attacking their blood. You can see that group AB, they don't have any antibodies because they have both. So an AB person can get group A blood, it can get group B blood, or it could get group O. Okay? Group O can only receive O because they've got A and B antibodies here. If you were to give them any of these antigens that you see in these top three, it would react with the antibodies of a group O person, and that would be detrimental. So an O person can be given to anybody, but an O person can only receive their own blood because of this antibody interaction. Now, of course, this is a basic introduction to this. We're going to touch on this again moving forward. And also, you're going to take a whole entire course called Immunohematology, where all of this is going to come together for you. So let's look at the properties of antigens. We also call them immunogens, but they're usually found as proteins or large polysaccharides. Rarely are they lipids. If you want to be a protein, or you want to be an antigen, you want to be a protein of high molecular weight. That makes you a fantastic antigen. You also have to have, and these are really important to know, I'll give you a hint right now, it has to be foreign, it has to be degradable, it has to have a good molecular weight, the higher the better, okay? There are haptens involved that are small but can bind a large molecule as well. They have to have structural stability, they have to be complex, and they have to be, again, foreign. That's the degree to which an antigenic determinant recognizes as non-self. The more foreign it is, the better off it'll be. All right, so we just talked about antigens and what those are. They're connected to all your cells, right? Let's talk about these antibodies that your body will produce to fight off foreign antigens. Antibodies are specific glycoproteins that are referred to as immunoglobulins. The function is to combine with an antigen to aid in the defense, okay, to help the body stay healthy. And after the antibody combines with the antigen, a secondary interaction occurs to dispose of those larger antigens, and that's known as complement that we'll get to in week 4. You can see here on the right-hand side on the top, there's an immunoglobulin. We're going to go over the different parts and pieces of those, and in lab we're going to actually build those. In order for antigens and antibodies to interact, we have to have specificity. This is the ability of a particular antibody to combine with a specific antigen. We see that the Fab molecule, or the variable portion, is the combining site that connects to the antigen, and we can find those on the top. Now, this is the Fab region up on the top, and my students call these the jazz hands. Okay, so it's fabulous. That's what the connecting point is. Fab are up on the top of the two hands, per se, that stick out on the top. Okay. We also have cross-reactivity. This occurs when some of the antigenic determinants of an antigen are shared by similar antigenic determinants on the surface of an unrelated molecule. Some antibodies can be directed against or react with another antigen as well. That's known as cross-reactivity. Now we're going to talk about affinity and avidity. Affinity is the initial force of attraction that exists between the Fab site of an antibody and one epitope or determinant site on the corresponding antigen. This is the strength of the bond, and it's non-covalent. Okay, so it's the initial force of attraction, or the bond. If you think of you have a strong affinity for another person, you may have a strong bond with that person. So that's kind of how I memorized that one. The next piece is antibody avidity. This is the functional combining strength with an antibody with its antigen. So when it comes to this, an avid exerciser has great strength. Okay, this is the binding strength. Each antibody unit has 2 antigen binding sites. Okay, I showed you those. Those are the Fab hands on the top. And the multivalent antigens binding to multiple combining sites of an antibody actually results in an increased bond strength. So let's look at each of these. You're going to be responsible for differentiating them. We've got 5 classes: IgM, IgG, IgA, IgD, and IgE. These differ from each other in molecular weight, sedimentation coefficients, and carbohydrate content. And they have different classes within them as well sometimes, especially this first one. First one's IgG. This is the most common. It's the major immunoglobulin found in the serum. About 70 to 75% of all immunoglobulin consists of IgG. Now, if you look at the top right-hand corner here, we have a molecule of IgG. It looks like the one that I just showed you 2 slides ago. It diffuses more readily into extravascular spaces. He can really get around. He neutralizes toxins or binds to microorganisms very easily. He's the only one capable of crossing the placenta. He occurs in the secondary response to an antigen that we're going to get to in a little bit. When IgG complexes are formed, complement can be activated. And we're going to learn about that in Chapter 4, but we need complement to finally come in and destroy those foreign antigens. We find IgG increased with infectious diseases, collagen disorders, and other hematological disorders as well. We do have 4 subclasses, Ig1 through 4. We see that IgG1 is most efficient at crossing the placenta, and IgG4 can be involved in allergies as well. Next one is IgM. This one is huge, okay? We've got, it's called a pentamer. There's 5 immunoglobulins connected together. It's 10% of the immunoglobulin pool. It's largely confined to the intravascular space because of how big it is. He's got a hard time moving out to extra tissues and stuff. He's produced very early in immune response, so he's the first one to come on, where IgG will come on later. He's very effective in agglutination. He can bind a lot of antigen. Okay, so he's pretty powerful. We do see this increased in infectious diseases, collagen disorders, and hematological disorders as well. Next one is IgA. This one kind of looks like a crab. Okay, so it's got like 2 immunoglobulins essentially stuck together. This is 15 to 20% of the immunoglobulin pool. This one we find in tears and saliva, milk, and intestinal secretions. So he likes to be in secretions. It's made by plasma cells located on your body's surfaces. It binds to a glycoprotein called the secretory piece in transport. So this one's very, very important for making sure you don't get eye infections or any other types of body openings. This one's increased in TB, collagen disorders such as rheumatoid arthritis, hematologic disorders, and some chronic active hepatitis as well. Next one's IgD. This one's found in the lowest concentration, less than 1%. It's very susceptible to proteolysis, and it's primarily a cell membrane immunoglobulin found on the surface of B lymphocytes. Oops, IgE is the last one here. This is a trace plasma protein found in blood plasma. It mediates types of hypersensitivity or allergic reactions, or allergies. Allergies sounds like IgEs, right? And anaphylaxis. So it can be increased if you have parasites. But the one big thing that it does is it binds to basophils, and it mediates the release of histamines and heparin. So if you've ever had seasonal allergies and you're sneezy and itchy, histamines and heparin are what make you sneezy and itchy. That's why you take an antihistamine to stop this process from happening so you don't have the release of all those histamines. IgG, like I said, is responsible for that. All right, so we talked very briefly in the beginning about the antibody structure, right? We talked about the Fab hands, the Jazz hands that grab onto those antigens. But there are other parts and pieces as well. So one region is concerned with the binding of the antigen. Okay, that's the variable region. And we also have a primary core that consists of amino acid residues called the constant region. The constant region contains 2 heavy chains and 2 light chains linked by disulfide bonds. Okay, and here's the picture. So here we've got the variable regions on the ends. Okay, these can vary from antibody to antibody. These long ones here are the heavy chains. Okay, and these small ones here are light chains. Okay, they contain 2 heavy and 2 light chains linked by disulfide bonds, like I just said, and there's 2 types of light chains. We've got kappa and lambda. The class and the subclass of the IgG molecule are determined by the constant region of the heavy type chain. It's really important to know. A typical IgG consists of 3 globular regions. We got the 2 Fab regions, the Jazz hands that we talked about over and over again, and we got the FC region, which is the stalk. Okay, this is kind of the stalk here. They're linked by a flexible hinge here in the middle, so these arms can move around and grab onto different antigens. Here's another picture. So here's that hinge that I was talking about. These are the basic labels of what you'll need to be responsible for. We have different types of bonding involved between an antigen and an antibody. It's exclusively non-covalent, but here's the type of bonds that are involved. Hydrophobic, hydrogen van der Waals forces, and electrostatic forces. We do have different types of immunoglobulin variants. Since immunoglobulins are proteins, they can function as antigens when used in an immunization. Also, they're found in 3 categories of antigenic determinants. If we have an isotype, it means there's differences in the constant region. And if you're an allotype, they have the same variable region and same constant region, but very little differences. I think they're all the same. That's how I memorized that one. In idiotype, there's differences in the variable region. And I always think idiots are variable people. So if you're trying to look for ways to memorize those, in the isotype, if you're isolated, you're in a constant region. Allotype, all the same. Idiotype, you're variable. So those are, nice ways to help memorize those. Now we're going to look at the primary and secondary antibody responses. An IgM antibody response proceeds in 4 phases after recognizing a foreign antigen. We've got the lag phase at the beginning where we can't detect antibodies. The body is starting to ramp up. The log is where the antibodies start to increase. The plateau is at the top where it starts to stabilize. Decline, the antibody is catabolized at the end. And I'm going to put a note in here. The titer is the quantity of a substance required to react with or correspond to an amount of another substance. So the higher the titer, the more antibody you have. Here's a picture of what I just mentioned. There we go. The primary response here, we've got the lag, we got the log, plateau, and decline. And you can see, wow, my fingers have a mind of their own today. The IgM increases first, and IgG comes in just after. So IgM's always in there to take care of stuff the first time you're ever exposed to something. So when you first get your flu shot, IgM's going to come in immediately, and IgG is going to come in with that memory. Then you're going to have a decline. Now let's say you got your flu shot. Now it's prime flu season and you're exposed again. Everybody's got the flu, but you got your flu shot. What's going to happen? IgM's still going to come in and play a role in the secondary response, but since your body made all that IgG, boom, it's going to come in and help kill off that flu antigen. We call this the amnestic response or the secondary response. It's that memory coming in to help take care of something. So again, exposure to the same antigen, it produces that antibody response. With the amnestic response, we see a much shorter lag phase, a longer plateau, and a more gradual decline. Usually IgM comes in, but IgG is primary, like I just said. Antibody titers, the more you're exposed, the higher the titer. So a titer means there's just more of them there. All right, lastly, we're going to touch on monoclonal antibodies. Antibodies that are monoclonal are cloned from a single cell. They have a great purity and specificity, and they're able to recognize and bind to a very specific antigen. The uses for these are identifying and quantifying hormones. We use them in some of our testing, for example, immunoassay testing. We add antibodies, which are very good at picking out the hormones and binding to them. We use it for typing tissue and blood. Okay, a lot of the reagents that we're going to use in immunohematology are monoclonal. You're going to be using monoclonal antibodies week 1, or you did week 2, to make a titer. You're going to identify clusters of differentiation. You're going to can use them for immunotherapy, and you can also identify tumor antigens and autoantibodies with these monoclonal antibodies. Here's how these are made. So what they do is they take the antigen, okay? Let's say we're doing an antibody for estrogen, okay? So we're going to take human estrogen and inject it in this poor little mouse here, okay? What they're going to do is they're going to take out the spleen, and they're going to look at these antibody-producing cells. They're going to mix them with the polyethylene glycol fusion and myeloma cells, and they're going to incubate, and we're going to see there's hybridoma cells that make one specific antibody per cell. And the best antibody-producing cells are cloned and expanded here and incubated. And then we're going to have an antibody produced in a culture supernatant that's going to be very pure. You don't have to memorize this process at all. just know that we're using some of these cells and the mouse to make this happen. Multiple myeloma is another issue where we see monoclonal antibodies that are actually produced by the human body. A myeloma protein is an abnormal immunoglobulin fragment, such as an immunoglobulin, that is produced in excess by an abnormal monoclonal proliferation of plasma cells. Hopefully in hematology you learned that plasma cells secrete antibodies. They're B cells that just secrete tons of them. In multiple myeloma, you have an abnormal monoclonal proliferation of those plasma cells. And what we find in testing is a protein called an M protein or an M spike. The proliferation of the myeloma protein has several bad effects on the body, including impaired immune function, very high blood viscosity, becomes very thick, and kidney damage can happen from that. My husband's grandmother actually died from this, and I remember what got her to the hospital was feeling very ill and having that high blood viscosity, which caused high blood pressure and kidney issues. Okay, well that concludes our lecture for Module 2. I'll hear from you next week.